many different molecules need to be able to go across the plasma membrane. Molecules need to be able to go into the cell, so like the raw ingredients, so oxygen, glucose, amino acids. Things need to go out like the waste products like urea, carbon dioxide, and also the products that the cell makes like proteins and hormones. To be able to do that, it needs to be able to go across the plasma membrane, which of course is selectively permeable. Being selectively permeable means that not everything is able to cross the membrane. And it's actually really helpful for a cell to have a semi-permeable, selectively permeable membrane to be able to control what goes in and out of the cell. That's one of the most important things that the cell membrane does, is it controls what goes in and out of the cell. And it does that in a number of different ways. We can divide it up into two key ways. We have got passive transport and, as you probably guessed, active transport. So it's called passive transport because no energy is required for the molecules to move across the membrane. And it does that because the particles move down the concentration gradient. So I'm going to explain what that means in a minute. But the alternative is active transport, and that requires energy. And it requires energy because it's movement against the gradient. Now, we can divide passive transport even further. I'm going to divide that into two key ways. Diffusion and osmosis. So before we talk about the differences between those two, I want to remind you of some terms. First of all, we have solvent. The solvent is the liquid in which the solute is dissolved. So the solute could be a solid, it could be things like salt, glucose, could be a gas like oxygen. The solute is the molecule that is dissolved in the solvent. Before we move on any further, I also want to show you when I draw square brackets like this, that means concentration. It means concentration. Okay, now, so we've got diffusion and osmosis. They're both passive ways of molecules moving across the membrane. Diffusion is movement of solute. Okay, so it might be glucose, it might be salt, or it might be oxygen. So it's the solute that moves, and it goes along a concentration gradient or down the concentration gradient. Osmosis is the movement of the solvent or movement of water. So again, there's a concentration gradient, but it's a gradient of water along the concentration gradient. And we're going to look at these two in more detail now. Just before we do, we can divide diffusion one more time. We have simple diffusion, which requires no assistance at all. It's just purely a passive process, requires no assistance. So it's unaided. Now this happens for very small particles that don't have a charge. So for example, something like oxygen goes across the membrane unaided through simple diffusion. It simply goes straight through the membrane. But most molecules can't do that because they're too big or they have a charge, like a positive or negative charge. So for that, we need a process called facilitated diffusion. Facilitated diffusion requires assistance, and it requires assistance through something called a protein channel. And it's for larger molecules like glucose and charged ions. Like sodium ions, for example, has a charge, so it can't just simply go straight through. Okay, so that's essentially the model. Okay, so we've got two types, passive and active. Passive transport, we've got movement of the solute, which we call diffusion, which we can, there's simple diffusion, which goes straight across the membrane unaided, or facilitated, which requires a protein channel. And then we've got osmosis, which is the movement of water along the concentration gradient. So that's it in a snapshot. Now I'm just going to go through diffusion and osmosis in a little bit more detail. Okay, so diffusion first of all. So you've seen simple diffusion before, so I'm just going to show you what facilitated diffusion looks like. So with facilitated diffusion, we have these things called protein channels. So proteins that are embedded all the way through the membrane create a channel. So we have got a high concentration of solute on one side, so high concentration of solute, and we've got low concentration of solute on the other side. So there's a net movement of the solute through the protein channel until we get dynamic equilibrium, where it's the same, essentially the same concentration on either side. So that's called facilitated diffusion. It goes across the protein channels. So simple diffusion is, again, you've got the concentration gradient, but you don't need the protein channels. It goes straight through. With osmosis, I want to just talk you through a couple of scenarios here. Look, importantly, with osmosis occurs when there is a solute concentration gradient, but the solute can't cross. Okay, so it's movement of, so for whatever reason, the solute can't cross the membrane. So we have a situation where the water moves instead, and that's osmosis. So let's talk you through some scenarios. Okay, here's a scenario here where we've got a cell in fluid. And in this particular case, we have got a high concentration of solute in the water. So that's like salty water. So that means if we've got a high concentration of solute, that means we've got a low concentration of water. So inside the cell, it's opposite to that. So that means that we have got high water and low solute. Now the solute can't move. So the water moves from an area of high water concentration to an area of low water concentration. So that means in this particular case, the water is going to go out of the cell and the cell is going to shrivel. The cell will shrivel because water is leaving the cell, so the cell is going to shrivel up. This situation here, we call this type of solution that's got lots of solute, really salty, for example, we call it a hypertonic solution. So tonic relates to the solute, and hyper means high. There's a hypertonic solution. Let's talk about another scenario, which is the opposite. Now we've got a situation where we have got low concentration of solute and high concentration of water in the outside of the cell. So thinking about what's happening in the inside of the cell, it's the opposite. So we've got low water and high solute. So water is going to move along a concentration gradient from an area of high water to low water. So the water is going to go in to the cell, and the cell is going to swell up. This situation here we call hypotonic. Hypo meaning low. There is a third scenario, and this is what we call, I'll tell you what it's called, isotonic. Iso means the same. So, in a cell, we've got the same solute concentration, same water concentration. So, there's no change. There's no net movement of water. There's no change. That's called isotonic, and there's no change there.